Namaskar, I am Vijay Dharmapalan and you are watching Science Time, a show that brings you the latest developments from the world of science and technology. Let's first look at some of the exciting stories. Revolutionary gene therapy offers hope for untreatable cancers. Scientists have created a vagina on a chip. A 2-million-year-old ecosystem in Greenland uncovered by environmental DNA. Now let's see the news in detail. Scientists in the UK have used a revolutionary new type of gene therapy to treat a young patient with relapsed T-cell leukemia. The administration of the technique, a world first, has raised hopes it could soon help to tackle other childhood cancers and serious diseases. Eliza from Leicester had undergone chemotherapy and a bone marrow transplant to alleviate her leukemia without success. With no further treatments available, prospects for the 13-year-old were grim. But after receiving an infusion of donated T cells, altered using a new technology known as base editing, Eliza is recovering and has been in remission for 6 months. Bases are the language of life. The 4 types of base, adenine, cytosine, guanine, and thymine, are the building blocks of our genetic code. Just as letters in the alphabet spell out words that carry meaning, the billions of bases in our DNA spell out the instruction manual for our body. Base editing allows scientists to zoom to a precise part of the genetic code and then alter the molecular structure of just one base, converting it into another and changing the genetic instructions. The large team of doctors and scientists used this tool to engineer a new type of T cell capable of hunting down and killing Eliza's cancerous T cells. They started with healthy T cells that came from a donor and set about modifying them. The first base edit disabled the T cells targeting mechanisms so that they could not assault Eliza's body. The second removed a chemical marking called CD7, which is on T cells. The third edit was an invisibility cloak that prevented the cells from being killed by a chemotherapy drug. The final stage of genetic modification instructed the T cells to go hunting for anything with the CD7 marking on it so that it would destroy every T cell in her body, including the cancerous one. That is why this marking has to be removed from the therapy, otherwise it would destroy itself. If the therapy works, Eliza's immune system, including T cells, will be rebuilt with the second bone marrow transplant. If these are successful, it is hoped that base-edited cells could be given to patients affected by other types of leukemia and other diseases. This is one of the most sophisticated cell engineering so far, and it paves the way for other new treatments and ultimately better future for sick children, said immunologist Professor Wasim Qasim, one of the project leader. Now let's move to the next story. A team of bioengineers have created a vagina on a chip to mimic key features of the vaginal microbiome. This simulated organ chip, made from donated vaginal cells, allows scientists to study the complex interactions between the vagina cells and fluids, as well as testing treatments for bacterial vaginosis, a common microbe infection which affects around 30% of women. The research is particularly important as the human vagina remains severely understudied by scientists. Women's sexual health has historically been vastly underrepresented in the scientific community, particularly when compared to men's sexual health. To this day, consequently, our understanding of the vaginal microbiome is surprisingly limited. But as detailed in the study published in the journal Microbiome, the vagina on a chip successfully allowed scientists to study the interaction between changing levels of estrogen hormones and bacteria. It's the most realistic lab model of the organ to date, according to its creators. This walks, talks, quacks like a human vagina, says Don Ingber, a bioengineer at Harvard and the co-author of the paper. Ingber and his colleagues have previously created other organ chips simulating the human lung, liver, and intestines, among others. Now let's move to the next story. Scientists discovered the oldest known DNA and used it to reveal what life was like 2 million years ago in the northern tip of Greenland. Today, it's a barren Arctic desert. But back then, it was a lush landscape of trees and vegetation with an array of animals, even the now extinct mastodon. The study opens the door into a past that has basically been lost, according to the lead author Kurt Kaiser, a geologist and glacier expert at the University of Copenhagen. With animal fossils hard to come by, the researchers extracted environmental DNA, also known as eDNA, from soil samples. This is the genetic material that organisms shed into surroundings, for example, through hair, waste, spit, or decomposing carcasses. Studying really all DNA can be a challenge because the genetic material breaks down over time, leaving scientists with only tiny fragments. But with the latest technology, researchers were able to get genetic information out of the small damaged bits of DNA, explained senior author S.K. Willersleigh, a genetist at the University of Cambridge. In their study published in the journal Nature, they compared the DNA to that of different species, looking for matches. The DNA fragments suggest a mix of Arctic plants like birch, trees, willow shrubs, with one that usually prefer warmer climates like firs and cedars. The DNA also showed traces of animals including geese, hares, reindeer, and lemmings. One big surprise was finding DNA from mastodon, an extinct species that looks like a mix between an elephant and a mammoth. Let's move to Science Quest segment. The question for this week's Science Quest is: do painkillers work differently for different people? In short, yes. It is thought that there is some individual variation in the response to a particular painkiller, but pain is a complex perception. Sensation is influenced by so many factors, including variation in the type of pain, genetic factors, stress-related physiological responses, socio-cultural influences, our prior experience, emotional state, and the context it occurs. So it is difficult to attribute our response to a painkiller to something as simple as painkiller receptors alone. But these are likely to play a part. With this, we come to the end of this edition of Science Time. Keep sending your feedback and suggestions through email. Our email ID is indiascience@vigyanprasad.gov.in. We'll be back with more exciting stories from the world of science next week. Stay tuned to India Science.